We're going to move away from fossils now and talk about something that may bring us a lot closer to understanding fully the remarkable diversifications that have occurred in flowering plants. And the key theme for this lesson is going to be genome duplication. Now, the general concept here is really basic. It's a doubling of the whole nuclear genome of the plant, all the chromosomes in the nucleus all at once. It happens in a single event of mutation. Take yourself, for example. You already know that you are diploid, meaning that each of your cells carries two complete sets of chromosomes, one from mom and one from dad. The total number of chromosomes in the diploid set in humans is 2n equal to 46. If you were somehow to undergo a whole genome duplication, you'd have 92 chromosomes instead of 46. You'd also call yourself a tetraploid rather than a diploid. Tetra is the Greek prefix for 4. If instead of a duplication you had a triplication of your genome, your chromosome number would be 6n equal to 138 per cell, and you'd call yourself a hexaploid, hexa for 6. The term polyploidy simply means having bigger ploidy numbers than just your standard diploid. Okay, now this is a big change to the genome, and obviously the first thing you want to know is whether or not we have evidence that it actually happens and that a mutation of such magnitude can actually take hold in the species. Well, I can give you a quick yes with qualification. Humans have benefited tremendously from the recent polyploidizations of plant species that we depend on for our food. From the very first time that humans isolated various plants from wild stock to use in our gardens, we've effectively managed their evolution into the plants of modern crops. And we did so by selecting the largest plants with the largest flowers, the largest fruit, and for grain crops like this wheat, the largest seeds. Now, when polyploidization occurs, one of the consequences is excess, exaggerated plant size, flower size, fruit size, etc. Like all mutations, this occurs only very, very rarely, but when it does happen in a food plant, maybe you find that one of the plants in your garden produces ears of corn that are twice as big as normal. Well, obviously you're going to save the seed from that plant for next year. In this way, polyploid mutants tend to get immediately selected as seed stock, and through subsequent crossbreeding and selection, you can end up with things like the modern market strawberry, which is an octoploid and enormous in comparison with wild diploid strawberries. In fact, if all of the recent human-managed polyploidizations of crop plants were to somehow revert back to the original diploid state, the Earth's human population would, after a generation of mass starvation, be reduced to a tiny fraction of what it is today. But this might be a special circumstance. After all, a human is there to selectively breed the polyploids. The question remains as to whether or not a polyploid mutation would have a chance in a wild population, that is, without the human there to snag it in order to breed bigger fruits, flowers, and seeds. Well, the short answer here is again, yes. You probably know that in recent years, we've sequenced the entire human genome, as well as the genome for a few other species, bacteria, plants, animals, and fungi. Basically, all the major domains of life have whole genomes published. What you're looking at here is a mapping of just one small segment from one of the chromosomes of the cress Arabidopsis thaliana. Now, the concept behind our method for detecting past events of genome duplication is really pretty easy to understand. What we do is we look for chromosomal segments, stretches of several thousands of bases like the one you're seeing here, that bear similarities with other parts within the same genome. Similarities that are so strong they can only be explained by them starting out as having been the same chromosome, that is, at the time of duplication. Now, it's also possible to estimate how long ago the duplication took place by looking at the evolved divergence between these matching chromosomal segments. If you take matching segments from a duplication event 120 million years ago, you expect to see a much greater degree of differences compared with what you would find in segments that started off as identical only 12 million years ago. Now, we've now looked at the genomes of many species, and when it comes to past polyploidizations, the findings are pretty astounding. So there are two groups that sort of stand out as having been especially affected by whole genome duplications, and one of them is flowering plants. The other is vertebrates. Most vertebrates, you included, bear evidence of having at least 2 rounds of whole genome duplication. Now, all the bony fishes, including everything from guppies to sailfish, have undergone a third fish-specific whole genome duplication. And there are even a few groups of fishes like the minnows and the trouts, which we're looking at here, that have undergone their own taxon-specific 4th genome duplication. Now, is it a coincidence that these 2 groups, bony fishes on one hand and flowering plants on the other, they stand out as both having had the largest number of genome duplications and also being the most successful and diverse out of their respective groups? You already know that flowering plants dominate the world's plant diversity. Now, the bony fishes similarly far outnumber all of the other vertebrates in terms of species diversity. Okay, so now let's think about how a whole genome duplication, or WGD for short, might actually contribute to higher rates of diversification and evolution. So just doubling the total number of chromosomes that are there in the nuclear genome, it's not going to add any extra diversity. We're just getting extra copies of genes that are already there. And in the short term, there's going to be a cost to managing this greater bulk, twice as much DNA. And so it's really unlikely for this to be selectively neutral, much more likely for it to be bad. Now I'm going to walk you through a sequence that leads to the extremely unlikely outcome of having a whole species convert from a diploid state over into a tetraploid state. And I can't emphasize enough that this is something that happens very, very, very rarely, but it does happen. Okay, so here's our little walkthrough. Okay, so use your imaginations here and imagine that this is a population of diploid organisms. And I'm going to put a single polyploid mutation in here. And so this organism right here is now polyploid. And I want you to think about, well, what's going to happen to that polyploid? Is it going to increase? Is it going to be dying out? What's the likelihood that over time this polyploid mutation is going to gradually increase and eventually take over the entire population? Is it a given that this is going to happen? Well, no, absolutely not. We've already said that the polyploid is likely to be worse off as far as fitness goes compared to the original diploids. If we're talking about an animal here, there's an extra difficulty, which is that this polyploid is now genetically incompatible with the original diploids, right? If this guy happened to be 4N and these guys are 2N, if they were to cross, you would end up with a 3N offspring, which is going to be sterile. In effect, these guys are genetically isolated from the original species, and therefore our polyploid mutant couldn't mate with a diploid original. Okay? So you would actually have to require 2 polyploid mutations to occur at the same time, and that's incredibly rare. Okay? So plants, in this respect, have a significant advantage over animals, which is that plants are often, if not usually, hermaphroditic. So that problem of finding your first mate is not going to be as much of an obstacle for plants as it is for animals. But in any case, even if we're just talking about a plant here, by far the most likely outcome is no change in the population. The organisms were diploid to start with. We had the polyploid mutation. The polyploid mutation died out, and then we're basically back to square one, diploid again. But this is really like any other type of rare mutation in the population. The most likely fate is for it to be eliminated. Okay. Now, on extremely rare occasion, a polyploid is going to get really, really, really lucky and end up taking over the whole population. It's not a new polyploid mutation anymore, so I can get rid of this. And what we're going to be talking about for the rest of this lesson is not how the polyploidy takes place or how it takes over the population. You can study this in a different class. It would be in the field of population genetics. Our focus this time is going to be on what happens in the aftermath of the whole genome duplication. In other words, we've already gotten to this point. We've overcome that first very substantial hurdle. This is almost never going to happen, but now that it has happened, what comes next? And more importantly, how is this going to be related to a rapid rate of diversification? So obviously there's a lot of genetics that I'm glossing over here. The history of our thinking about genome duplications and their impact on evolution started back in 1970 with Susumu Ono's seminal book, which was Evolution After Gene Duplication. And so we're talking about a 45-year history that's gotten really, really interesting just in the last decade. So I'm exercising quite a bit of constraint to not talk about all this other stuff. My objective here is really only to examine the potential reasons why there might be a period of high evolvability following a polyploid event. Okay? I'm cutting things down to the bare bones here to make this connection. So let's take a species that's recently become a tetraploid. What's going to happen to it over the next few hundreds of thousands of generations? Well, obviously there's a lot of excess in this tetraploid genome. There are a lot of genes and even whole sections of chromosomes, and maybe even whole chromosomes that are unneeded. Or maybe they're just barely needed, and with a bit of modification, they would become completely useless. Okay? Now, there are mutations that can remove unnecessary parts of the genome, and over the course of time, they're going to have the effect of pruning away those useless parts in the same way that I might prune away some unproductive stems in a citrus plant that I have in my front yard. Now, this pruning metaphor is imperfect, because when I do the pruning of my tangerine tree, there's a real deliberateness to my pruning efforts. I decide which are the parts that I don't want, and I only remove that material. When a mutation snips away at a genome, the removal of DNA is totally at random. When a mutation happens to cut out a crucial bit of DNA, in other words, a piece that's really needed, that mutation is going to fail. And only those mutations, only those cuts that happen to leave the important parts intact will be the ones that have any chance of surviving. Now, over the same period in which the genome is getting randomly pruned down to a more manageable size, the duplicated genes that were retained are evolving to work better in this new context of tetraploidy. Sometimes the duplicated copies of the genes might continue to function exactly as before, and this gives us an amplification of gene expression. Think about that oversized strawberry. Why would a polyploid tend to be bigger? Well, some gene products that were in lower dosage in the diploid are now there in higher dosage, and this permits an increased synthesis of the gene product. Other times, the duplicated genes can diversify, with one copy maybe retaining its old function and the other copy evolving to take a slightly different or maybe even a very different function. Now, this calls for an example from the real world to illustrate how a modest change in gene function can actually lead to something positive. Now, there are lots to choose from, but as usual, my favorite ones come from the fishes. Now, think of a set of genes that's responsible for normal muscular function. They produce the sodium channels that normally generate electrical signals that cause muscles to work. Fishes need these sodium channels. They need these genes for all of the things that they need to do that involve muscles. Swimming, eating, breathing. So these genes are pretty important. Okay. Now remember that being vertebrates, fishes already have duplicate sets of their genes. But in the case of bony fishes, there's a whole extra level of duplication. So think about how an extra set of these sodium channels could be modified. Maybe to generate electrical impulses with a much higher amperage. And maybe this is going to happen in special electric organs that the fishes could use in some way, maybe to stun prey. Well, this is exactly what we're seeing in the electric eels that live in South America, as well as a few other weakly electric fishes living in Africa. We've already talked about the pruning down of genome size, and now we've talked about duplicate copies of genes diverging in function. There's also this rewiring of the genetic network that's going to be needed. For example, in the case of the electric fishes, we need to have that extra set of sodium channels activated and expressed in different locations, and therefore with a different control mechanism relative to the sodium channels we have going on in the regular muscles. And this is going to require modifications to an array of other genes that control the development and expression of the genes that are diversifying. And I know this sounds complex, but it's really kind of manageable. Now let me try to illustrate this process, starting with the duplication of a genome that originally consists of only 2 chromosomes. The 4 chromosomes you're seeing here is because it's a diploid. Once you duplicate it, it's going to look like this. And so you've got 8 total chromosomes now. So for simplicity's sake, let's call the lower tier of chromosomes here the duplicates, right? And I'm going to start by identifying all the parts of the duplicates that are desirable to be retained. I'll mark them with yellow, and I've also changed the color of the original set of chromosomes so that we can distinguish them from the ones we're calling the duplicates. Okay? But in reality, it's not like one set is the master and the other the duplicate, right? Both sets are under the same type of evolution. It's just for simplicity's sake that we're kind of identifying duplicate and master here. So again, all these yellow genes are the ones that are retained. The blue parts, the dark blue parts, are really unnecessary. Now, if we look at the second chromosome here, it looks like almost the entire chromosome is unnecessary except for this one gene that we have here. We can't simply ditch this chromosome altogether because we have this one gene that we want to keep. However, if there were a chromosomal rearrangement that took these genes here and moved them to a different part of the genome, maybe up here, right? If that were to happen, then these chromosomes could be totally eliminated. And so now let's see what it's going to look like if we actually make those changes. The fourth chromosome is gone. We've taken the one part of the fourth chromosome that we wanted, we put it on the end of the second chromosome, and we got rid of the fourth chromosome at that point. Now, it's also possible to undergo other kinds of changes that could result in the removal of, say, maybe this section right here, right? Just simply take it out. And so if we do that, the chromosome is going to appear a bit shorter, and I'll show you what that looks like. So this kind of alteration of genomic organization can result in a much smaller genome, or it might also result in relatively little size reduction. What happens to the genome kind of depends on the nature of the mutations that occur. So now let's think about the control of genes, how they get turned on and turned off. Now, for each gene in a eukaryotic organism, there are potentially several different factors that have to combine in order to activate the gene, maybe a different set of factors that combine in order to turn a gene off. But again, we're going to keep these simple here, and let's say there's a gene right here whose product is responsible for turning on this gene, which is now there in 2 copies, so it's also turning on the same gene in the duplicate chromosome. Okay? But in the process of subfunctionalization, that's the divergence that's going to occur between the 2 copies of the gene A and B, we might want one copy to be expressed in a different situation. Okay. So what we're going to do here is we're going to cut off this line. So this copy of the gene is no longer going to be under the control of the original control switch. The control of this gene here is going to be carried out by a different portion of the genome, maybe a gene down here. And so when we do this, the genetic network has now been altered. We've rewired this genetic network. So when I talk about rewiring, this is what I mean. Keep in mind that there are potentially thousands of gene copies that are undergoing this kind of evolution at the same time that the genome as a whole is being snipped away by that random gardener. So there's a mind-bogglingly large number of possible directions that evolution could be taking, as well as a near inevitability that some of this evolution will be occurring in a recent polyploid, given all that genomic excess and the potential paths for genomic reorganization. The species and its genome is going to be evolving some way in the next 100,000 generations, and it's pretty likely going to be substantial. We just don't have any clue about exactly what's going to be happening. Okay, so now the issue of how genome duplication relates to the process of speciation, or new species origination. In the standard model of speciation, also known as allopatric speciation, things start off with a single species that's been split into geographically isolated populations. Okay? So all 4 of these populations of ducks are the same species initially, but they're geographically isolated, each in its own lake, and moreover, they only reproduce with members of the same isolate. Okay? Each population, each isolate, evolves along its own independent path, and given a long enough period of isolation from all the others, each population will evolve, and it eventually reaches a point where it's unable to interbreed with the others, even if they're brought back to the same habitat. Keep in mind here that it doesn't matter what the isolates look like. They might have changed drastically or not at all. If they can still interbreed when brought back into the same habitat, then they're still the same species. Speciation is not yet completed. If you bring them back together and they can't or won't interbreed, then speciation is complete. We tend to think of speciation as a special class of evolved changes. Instead of thinking about things as being either adaptive or non-adaptive with respect to the local environment, in the case of speciation, what's important is the evolution of some kind of species isolating mechanism. It's something that will occur at some point over the course of this geographic isolation, but we can't predict when it will occur or even what form the isolation will take place. Now the key for us is that species isolation or speciation is going to occur eventually, and this basically means that it could be sooner or it could be later. Now, if the original species was a recent polyploid at the time of geographic isolation, there is good reason to expect species isolation to be reached sooner. Each of those local populations will be paring down their genome and rewiring their genome in its own way. And this kind of genome evolution is just a very good way for the individual isolates to become reproductively incompatible with other closely related populations, and also to do so quickly relative to what would happen in the absence of the initial polyploidization. With this in mind, I find it to be a tantalizing thought that these explosive radiations of angiosperms, starting in the early to middle Cretaceous and continuing to the present day, could have been facilitated by whole genome duplications. That makes a pretty good story, doesn't it? What else do you need? That's right, evidence. We already have a general concordance of there being both lots of whole genome duplications in angiosperms as well as a high speciation rate. The question now is, can we connect the two phenomena within the phylogenetic history of angiosperms? Now this question is quite a bit harder, but with the state of our present knowledge, things are looking a lot like a yes. With a recent flood of available data on whole genomes, as well as these EST studies, expressed sequence tag studies, that have provided a large amount of sequence information for large proportions of genomes, people have made some interesting findings. Now, as with all of science, there are some limits to the confidence in our conclusions. And here in particular, there's the caveat that our skill at detecting and evaluating whole genome duplications from the various formats of data is still imperfect. And yet, there are a handful of conclusions that can be drawn with a pretty high degree of confidence. 1. Polyploid events are common in plants, and especially the angiosperms. We used to think that only some plants, maybe 50% of all species of plants, had histories of polyploidy. Now it's no longer a question of whether or not a plant species is a polyploid or not, but rather how many polyploid events has it gone through in its history, and how long ago were these events. 2. All flowering plant genomes bear strong evidence of past polyploidy, with one exception, Ambarella trichophyta. This is one of the few species for which we have a complete nuclear genome, and there's a remarkable absence of evidence for whole genome duplications for Ambarella. This is another good example of a piece of information that can help us place the timing of a duplication event. In this particular case, it suggests that the whole genome duplication shared by all the other angiosperms must have occurred after Ambarella split away, but before the common ancestor to Nymphaeales and the rest of the angiosperms. 3. Whole genome duplications seem to occur near to the base of those pulses of diversification. A genome duplication is not, however, a guarantee that rapid diversification is going to take place, but there is a statistical relation between genome duplications and bouts of explosive diversifications. 4. There's a cluster of whole genome duplications in several different taxa, dating to the same period around 65 million years ago. This is roughly synchronous with the Chicxulub bolide impact and immediately preceding another major bout of radiation in several angiosperm taxa. Now the general idea here is not that the meteorite created the polyploidization or caused the polyploidization, but rather that the stressful times following this destructive impact event opened many more ecological opportunities. And this could have made it easier for polyploids to persist, since under normal, non-apocalyptic times, there would have been greater competition from existing flora, and under those circumstances, polyploids might have had a much lower rate of success. Now, if you look at points 3 and 4, these are observations that actually support the idea that whole genome duplication is a factor that contributes to the rapid diversification, at least the ones that we've seen in the angiosperms. But this doesn't mean that it's the only factor, right? There's no reason that we have to have only a single cause to the exclusion of all the others. In a previous lesson, I mentioned that while dinosaurs didn't seem too plausible as a driving factor for angiosperm evolution, insects, in particular herbivorous and pollinator insects, would have been strong contenders for biological partners in this massive diversification. Now, all of my sources for this series are presented in an annotated bibliography accessible from my YouTube channel. But for this video especially, I wanted to identify at least a couple of sources that stand out from the dozens that went in the development of the script. Now, the foundational work on gene duplication as an important phenomenon for evolution was Ono's book, Evolution by Gene Duplication, published in 1970. Two other studies, Tank et al. In 2015, Van Este et al. In 2014, they provided the impetus for my decision to include this topic at all. It's because of their findings that directly connect whole genome duplication to angiosperm radiations through their history, and particularly at the Cretaceous-Paleogene boundary. To summarize, one possible mechanism underlying the explosive radiation of flowering plants involves the phenomenon of whole genome duplication, which is both well documented in plants by recent genomic studies and also well enough understood to support a connection between genomic evolution in the aftermath of a polyploidization and accelerated rates of species diversification. We're not done with this story. One of the things we talk about later is the genetics underlying plant development, like the genes that cause plants to induce flowering and how plants make a flower out of the parts that were ancestrally just regular vegetative leaves. Now, this family of developmental genes is one that has become highly complex in flowering plants, owing largely to this past history of genome duplication events.